Okay, good morning. My name's Jeffrey McCowich. I'm a paediatric oncologist, a children's cancer doctor at the Children's Hospital in Westmead, which is in Sydney, Australia. And today I want to take you through uh, a paper that's been published recently on the treatment of neuroblastoma. So let me take you to that and get rid of my face and show you what's going on. So first off, we'll just go to the desktop and just that's me. That's my name and title there. And yeah, I'm at the Children's Hospital Westmead. That's like the biggest children's hospital in Sydney, Sydney in Australia. So now I'll show you what we want to talk about today. So there's a paper that's been published recently. And this is the paper. Here it is. It's in Nature Medicine, very, very prestigious medical journal. You get a paper published in Nature Medicine, it's pretty much a career maker. You can see it's been published just in August 2025. And one of the things I want to stress from the start is that I'm just here showing you what the Nature Medicine people put up for free on their website. I haven't logged in, I'm not using a paid subscription. This is just the information that they have already put out there that's free for you to get access to. And so I hope they don't mind me now taking people through it in a sort of plain English, I guess. That's what I want to do. And it's an article in high risk neuroblastoma. So that means that's the one that needs the most heavy treatments these days to try to cure the cancer. And we've made a lot of progress, but we need to make a lot more high risk neuroblastoma. And in particular, this group, and they're an Italian group, and here's the senior author, Francesca del Buffalo, and Franco Locatelli is the first author. It's a big Italian group, and they're reporting the use of these CAR T cells as treatment of neuroblastoma. And in particular, CAR T cells that target this chemical, GD2, which is a chemical on the surface of neuroblastoma cells. Okay, now, what I want to do first off is just explain to you about CAR T cells. Now, if you go and just Google CAR T cells and CD19, you'll find all sorts of articles and things. And here's one that has a nice picture to show you. This group's News Medical Life Sciences. Okay. Anyway, they've got a good picture that I want to show you to explain what a CAR T cell is. And CAR T cells have been most successful so far in the treatment of leukemia, particularly a type of leukemia in children called acute lymphoblastic leukemia, which is the commonest leukemia in children. This picture shows you up the top here, this peachy coloured cell. That's the leukemia cell. And one of the things in this type of leukemia is that there's a chemical on the surface of leukemia cells called CD19. And there's a picture of a CD19. And it's a normal chemical on all sorts of blood cells, well, different types of blood cells. But it's on the surface of the leukemia cells. So it's right there on the surface. It's a good thing to try to attack. Now, this is the CAR T cell. I'll just see if I can make this a bit bigger for you. And I think I can. Yep. Okay. So this is the CAR T cell. Now, first of all, T cells are one of our normal white blood cells we have running around our bloodstream. We've got neutrophils and basophils and monocytes and B cells and T cells. And T cells are important in our immune system, and they kill off viruses in particular, and sometimes they kill off tumors that are trying to grow before they ever get too big. And they reject organ transplants if we don't suppress them. That's what T cells do. So they're an important part of the immune system. Now, CAR T cells are T cells from the patient with leukemia. So they collect T cells from the bloodstream of the patient with leukemia, and they're just normal T cells. Then they take them to a very space age sort of lab, very sterile, very clean, very super high tech lab, and modify them to introduce into them a DNA, a gene, into the DNA of the T cells. And then that DNA has the recipe for the cell to make this thing that's here on the surface of the cell now. So that wasn't part of a normal T cell. That's something that, because the cell's been modified in the DNA, it's now got the recipe to make this structure and to put it on the surface of the T cell. Now, let me take you through this structure. This structure that now sits on the surface of the T cell has on the outside what's pretty much an antibody against CD19. So antibodies, we have antibodies that help us fight infections and things like so. To stop you getting tetanus, you have a vaccination, so you've got an antibody against the tetanus toxin, right? So we have antibodies against all sorts of things, and chickenpox and viruses and all sorts of things. Well, this is an antibody that's been manufactured to be anti-CD19. So that means when these CAR T cells are then given back to the patient in their vein, they can circulate through the body, and the CAR T cells have this antibody to attach to the target on the leukemia, the CD19 that's on the leukemia cell. And that's what they do. Now, on the inside, the antibody binds to the CD19, but then through some complicated biochemistry here, that activates these chemicals on the inside of the T cell. And these are the chemicals that tell the T cell to kill. And more than that, they tell the T cell activate, divide, multiply, make many more copies of yourself, and kill that target. So they're the activation domains, the co-stimulatory molecules on the inside of the T cell. So there's the ectodomain, the bit that's on the outside that binds to the target, the antigen, CD19. Then there's a linker, a transmembrane domain, and then there's these chemicals on the inside that tell the T cell, get active, kill the target, multiply, make many more copies of yourself, and kill the leukemia. And that's how a CAR T cell works in leukemias that have CD19 on the surface of the cell. By the way, here they've acknowledged where they got this picture from. Doctors Britton and Oliver, 2019. Okay. I hope they don't mind me showing you the picture. I don't think they would. And it is free on the internet anyway. So that's CAR T cells. And the CAR bit stands for chimeric antigen recognition. Chimera is a mixture of things. Okay. So this is a mixture of some antibody things and some T cell activation things. And that's why it's called a chimeric antigen receptor T cell. And these have been huge in leukemia, made a really important development. Now commercially available and covered by the government in Australia and elsewhere to provide this treatment to children with leukemia in the right setting. So that's a chimeric antigen receptor T cell. Now, neuroblastomas, on the other hand, another form of childhood cancer, they don't have that CD19 on their surface. It's just not there. But they do have some other chemicals that are on their surface, And one of them is GD2, which is a big long chemical name. Don't worry about it. GD2, it's a normal sort of chemical in the body, but it's particularly found on neuroblastoma cells. And one of the biggest developments in neuroblastoma treatment in recent years was the development of dinutuximab. Now, that's an antibody against GD2, and that's in very routine use in the treatment of high-risk neuroblastoma and has been a big breakthrough in neuroblastoma across America, Canada, Europe, Japan, Singapore, I expect, China, Australia, lots of places. It's a big deal to give, but it's made a big difference. So that's an antibody against GD2, dinutuximab, also called Qarziba, the European version of it, and or Unituxin, the American version of it. They're dinutuximab. Now, some other groups, various groups, have thought, let's make a CAR T cell against GD2. And by the way, people who have attacked GD2 with CAR T cells in some other diseases as well, melanoma, one of the brain tumors, some other diseases. But in particular, today we're talking about neuroblastoma. Now, what's going on with this paper is that this same group previously reported their results with their CAR T cell that they manufactured and treated children with neuroblastoma. And they've already reported results from the earlier part of this study. And those results were published in the New England Journal of Medicine back in 2023. So there you see Dr. Del Buffalo again there, and a number of the other authors there, and another 28 of them are there, all the way down to, and there's Dr. Locatelli there. So they already reported that in the New England Journal of Medicine. And again, I haven't logged into the New England Journal of Medicine. This is a freely accessible version of the paper that you can get by searching at the nejm.org, and then you can look for, if you look for Buffalo, you'll probably find it if you search under the authors, but it's free and out there. And so I hope they don't mind me just showing it to you today. But they previously reported this paper. Now, what happened in this paper was they had children with neuroblastoma and they were high risk neuroblastoma. So here we are, they had 27 children with heavily pre-treated neuroblastoma, 12 with refractory disease. That means the cancer won't go away with chemotherapy. They can't get rid of it. 14 with relapse disease. That means they treated it, maybe the scans were clear or almost clear, and then it grew back again. And both of those are very serious situations. That's when we're really worried. If we can't clear the cancer, refractory disease, or if it grows back after treatment, that's a really serious situation. And one with a complete response at the end of first line therapy. So that's someone who's had their treatment for high risk neuroblastoma, and it sounds like the treatment has gone okay, and the scans are all clear, but nonetheless, they put them into this study. Okay. So, they collected these blood cells from the patient, lymphocytes, they're called, and they generated autologous, that means it's from the patient, GD2 CAR T cells, and treated the patients. Now, the other thing it mentions here, by the way, this inducible caspase-9 suicide gene. When you give CAR T cells, you have to worry, as well as attacking the cancer, what if they attack some other part of the body that also has GD2 or CD19 or whatever your target is? And so what they've done here is they've put in this suicide gene so that if the CAR T cells caused a big problem, big toxicity, they would have a way to just eradicate the CAR T cells. They could give this other medicine, and they did give it to a patient, which would clear the CAR T cells and get rid of them. So that's not good as far as killing the cancer, but if they're causing big problems, you need to get rid of them. Yeah, there you go. One patient, they activated the suicide gene and rapidly eliminated the cells. So that's what they gave. All right. So they previously reported this. And as just to rip through this, there was 27 of them, no failure to generate CAR T cells. So they were able to make these CAR T cells on the patient, which is a high tech, extremely expensive process of, um, stimulating the T cells and then using an artificial virus to deliver that little bit of DNA. That's called a vector. I think it was a retroviral vector. That's a type of modified virus that just delivers the gene and doesn't do anything else. And that's, one of the big things is to generate that vector. And there's a lot of science in that, the shape of that antibody, the shape of that linker sequence, the shape of those co-stimulatory domains. It's all super high tech and different people have different ways of doing it. And people have tried this and they've tried that. And a lot of it's covered by patents because people spend years and years generating their version of it. And uh, it's uh, it's complicated and an area of ongoing intensive research. But anyway, they were able to make the CAR T cells and they tested three dose levels of these CAR T cells. So 3, 6, or 10 by 10 to the 6, 10 million then, CAR T cells per kilogram of body weight. So what they're doing here is first they have to prove that it's safe to give these cells. And so they start at a lower dose. So the first few patients got 3 million T cells per kilogram of body weight. And then when they showed that that was safe, they would have gone up to 6 and then up to 10. So that's why it's called the phase one portion. That's a dose escalation phase. They're giving the first group a smaller dose, then a medium dose, then a higher dose until they get to a dose that they think is as high as you need it to be. And then that's the phase one portion. And then they said, all right, so the recommended dose for the phase two portion was the 10 million. So 10 million CAR T cells per kilogram. So that's good. They've worked out a dose you can give, and then they've gone on to a phase two portion and given 10 million of these CAR T cells per kilogram. All right. And they report that 74% of patients got this cytokine release syndrome. That cytokines are chemicals in your blood. They're sort of part of the immune system. And when you rev up the immune system and T cells and kill other target cells, you get these cytokines. They're chemicals that run around your body. And the cytokine release syndrome might be with high fevers or blood pressure changes or rashes or all sorts of things. It can be very serious, but it was mild in 19 of the 20. That's a big thing in the leukemia CAR T cells, the cytokine release syndrome or CRS. It's well described, and it's one of the things. If you're giving CAR T cells, you need to know how to manage CRS. Anyway, it was mild in 19 out of 20. And in one patient, they had to give the suicide gene activation to kill off the CAR T cells. Next, they showed that these CAR T cells did in fact expand in the body, right? That's good. We want to give, they gave their 10 million per kilogram, and you want to see that their numbers, measuring a blood count of CAR T cells, that the numbers expanded, and that was what they wanted to see. Um, they were detectable in the blood. In 26 out of the 27 patients, well, they eliminated them in one up to 30 months after the infusion. Median persistence. So on average, they lasted about 3 months, but 1 to 30 months. And they said that 17 children had a response to the treatment, meaning the scans and measures of the neuroblastoma improved. They had an overall response of 63%. 9 had a complete response. That means no trace of neuroblastoma could be seen on the scans. 8 had a partial response. A partial response normally means something like about a 50% reduction in the size of tumours. So in patients with refractory disease, the chemo wasn't working, or relapse disease, that's an exciting result, and that's why you get in the New England Journal of Medicine, very prestigious journal. And particularly in the patients who got that higher dose, the top dose that became the recommended dose, the 10 million, the 3 year overall survival was 60%. So if 3 years later, 60% of the patients who got the full dose were surviving, and event-free was 36%. So some were surviving, but the neuroblastoma was active again. So that was pretty exciting, and that's why you get in the New England Journal of Medicine and have the whole world talking about it. So we congratulate these authors and the New England Journal of Medicine. And if you do get a subscription to the New England Journal of Medicine, there's this quick take thing, that'll take you through it. There's all sorts of other things, and you can get the full article. Actually, I think you can get the full article now. Sometimes they do this 2 years later. So you can actually read the full article there, but I might push my luck if I try to do that with you, except to mention that it was a retrovirus. See? And by the way, before you give CAR T cells, you give high dose chemotherapy to sort of knock down the body's remaining T cells, sort of make room for the infusion ones. So they gave this fludarabine cyclophosphamide chemotherapy. That's high dose chemotherapy to knock down the body's immune system. So when you give these cells back, they will sort of have space to grow, and that makes them engraft better. Cyclophosphamide, of course, is used in treatment of neuroblastoma. As many of you will know, fludarabine, not as much. It's more a leukemia drug, but that's not why we're giving it here. But yeah, all the detail is there, but I won't take you through all of that. Okay. So that's exciting. Congratulations to them and to the New England Journal. And if you want to get more, you might need to create an account and so on. And over here, you'll see there's an editorial. That's what medical journals do sometimes. They'll get a couple of eminent people who weren't part of the publication team to say, well, what do you make of this paper? And they're two eminent physician scientists, and they've written an editorial. But again, you click on that, um, you need to have the right level of access to read it. And then you'll see here some letters people have written in after it was published in April 2023. Various people have written letters in, and that may or may not be available. Oh, yeah, here's someone's. Again, you need the proper account to read it, but here's someone who's written in and said, well, this is what we think. And I don't know what the letter says. Did they say we don't accept your data, or we think it's fantastic, or uh, some scientific thing they will have written about, and then the authors will have responded, and so it goes. All right, so that's the article from 2 years ago, first describing these GD2 CAR T cells. So now we come to the Nature Medicine paper, which again is freely accessible on the internet, and there we are, the same first author and senior author, but they've reversed their position. Now, what they've done now, they're reporting. Here's this thing called the abstract, it's the summary. And they said, yes, we showed encouraging efficacy, and we now present the final results in all 35 patients enrolled in the trial. So remember when they published it before, there was 27 patients in the trial part of it. Now there's 35 enrolled in the trial part of it, so another 8 patients. And then in 19 additional children who were selected with the same criteria of the trial. So those similar sort of patients with neuroblastoma that was refractory or relapsed, but they weren't in the official part of the trial. They were treated in a hospital exemption setting. That probably means that their hospitals said, okay, you've finished that trial and all, but the results are such that it's okay to keep treating some more patients. And so there's 19 additional. So that makes a total of 54 patients that they're now reporting. And they take you through what they're going to tell you about the endpoints, safety, the cell dose, the activity of the response rate, the activity of the cells, and the 5 year overall survival and persistence of the cells. So they say no new safety signals. So the same things they reported in the New England Journal are about what they saw here. They did see some immune effector cell associated neurotoxicity syndrome. That's called ICANS. We see that in the leukemia CAR T cells as well, immune cell associated neurotoxicity syndrome. And that can be things right from agitation through to tremors, through to confusion, seizures, even more serious things. So they saw that, which is because of the immune system being revved up in some way and affecting the brain. And so they rapidly controlled it by giving that drug to activate the suicide gene. That's the name of the drug there, Rimiduside. That is just a drug that activates this suicide gene in the T cells and clears them out of your body. It's unfortunate because you want them to kill the neuroblastoma, but if they're causing trouble, you have to get rid of them if it's severe trouble. They again say 10 million cells. And here we go. The ORR is the objective response rate of the patients in the trial, right? So remember, there was a total of 35 in the trial, but three of them didn't have disease on their scan. So there's 32 where you can say, did the disease improve? And the objective response rate, so an improvement on the scans or on the bone marrow or the urinary catecholamines, however they were measuring the disease, there was a reduction in 66%. And the complete remission rate at 6 weeks was 37. So a complete remission, that means you can't see the tumor anywhere at 3 months and 6 months, 37, 34, and 40%. Okay. The CAR T cell persistence, more than 12 months in 64% of patients in the trial. So in the trial group, here's the updated figure, the 5-year overall survival. So if you looked at these patients 5 years later, 43% was still alive. And for such high-risk patients, that's showing that this treatment looks like it's delivering. All right, now they get here and they to this point, and now they're starting to add in those patients who were treated not on the trial. That's these extra 19 children on top of the 35 in the trial. So 38 of 54 had a low disease burden at the time. Okay, so meaning that the on the scans there were just small amounts of neuroblastoma that could be seen. That's what it will mean. They'll have an official definition. And they're considering them the main target group, not the patients with larger amounts of neuroblastoma deposits. And so there were 38, 8 of them had no evidence of disease at the time they got to the CAR T cells. Now, in the target population, the response rate was 77%. So in patients with a smaller amount of disease at the time of the treatment, 77% had an improvement. And the 5-year overall survival was 68%. Okay, remember it was 43% overall, but if you just looked at the patients who had clear scans or just lower levels of abnormality from neuroblastoma, then 5 years later, 68% of the patients were alive and 53% event-free survival. That means they're alive and the cancer hasn't become active again. So it hasn't progressed. It's either stayed absent on the scans or it hasn't got worse. Progression. That's event-free survival. So they make some other points. They talk about how, if you come to this and you've only had, say, the first line treatment of neuroblastoma or the second line treatment, that was better than the patients who've had multiple different attempts at chemotherapy. Here's something interesting. Better results were observed in patients who had those lymphocytes collected at the time of diagnosis. So what's going here is um, in many patients, they will have started treatment for the neuroblastoma, and it's only later in the piece, maybe if the treatment doesn't work, that they went back and collected the lymphocytes to make the CAR T cells with. They collected these T cells. So they've collected them in a patient who's already had a lot of chemotherapy. And so those T cells, lymphocyte, mightn't be as healthy and robust and able to respond But in certain other patients, they've collected them right at the initial diagnosis of neuroblastoma. So that's before those cells have ever had any chemotherapy. So that's interesting. And it's not something that, people are automatically doing and would deserve looking through the details of that and looking into it. Uh, but that's what they're saying. Anyway, so they conclude that these CAR T cells can induce durable remission. So what they've done really is extended the data they already had for us with 27 patients, now with, well, twice as many patients. And, the data seem to be holding up. That is, that they are continuing to see that this treatment looks like it's an effective treatment. So that's what the paper's about. And again, yeah, you'll have to access it if you want to get the whole article. And I hope Nature Medicine don't mind. By the way, here's something interesting. They've a publisher correction. Sometimes you publish a paper and then someone works out, oh, we said the wrong thing, or where there's a typo or something. So they've done a publisher correction. Well, I looked at the publisher correction, and then what they did, the original graphs, panel 4B was just a duplicate of panel 4A, and they'd left out, it looks like they've left out panel 4B, so now they've put it in. So that's not a major criticism or anything. Sometimes people say, look, sorry, we said the wrong thing, it was all wrong. They're not saying that. They just had their graphs mixed up or something, and now they've got them straightened out, so that's fine. So that's pretty exciting. It's a high-tech technology. Other people have tried this GD2 targeting CAR T cells, and this is the most exciting result, I would say, in neuroblastoma. Why is it so? That's really, what do they say? You really have to dig through the weeds to find out why has it proven effective here. But there's so many components of it that are different from one trial to the next. And for instance, like I said, what the design of the antibody is, what's in this spacer and linker, what cocktail they use to generate the CAR T cells, all sorts of things, which co-stimulatory molecules you can have here. There's first generation CARs and second and third. There's so many elements just in the CAR T cell design, in the CAR T cell manufacture. There's a whole lot of things that go into this that people who live and breathe CAR T cells, like me, uh, pore over and try to work out, well, why is this proving more effective than certain other GD2 cars? So it's certainly not something you can just order over from the pharmacy. Oh, send them over or something. This is a huge undertaking, and you need to be able to make CAR T cells, which we can do, by the way. We haven't made them yet for neuroblastoma, but we're working on things, but we are giving them for other diseases. You can go to ChatGPT. Here you go, and have a look at CAR T cell trials underway in the treatment of neuroblastoma, and there are there's GD2 CARs, B7 CAR T cells, particularly I think Seattle. This one's new Glypican 2 CAR T cells. I think that might be at the Children's Hospital of Philadelphia. PRAME, we're working on PRAME actually in childhood cancers and trying to develop these TCR engineered cells. You can't use CAR T cells for this one because it's not on the surface of the cell. It's on the inside. So there's other people doing other things, and it might be many of them might be getting away from retroviral vectors, which are a bit older. And for instance, in our trial in sarcoma, we're looking to use a lentiviral vector, which has some advantages. There's a whole lot of components of everything to plough through. These are huge undertakings. But I have to congratulate these Italians, this Italian group, for what they've achieved. And we'll continue to follow with great interest and hope that this can become more widely available and further enhancements to it all can be generated. But I'll stop there and I'll catch up with you next time. Thanks.